Check out all these Anubias plants that I just harvested from my ebb and flow system that's just over my shoulder here. Now these plants were part of an experiment that we were running to evaluate the differences between a few different growing conditions and find out what grew the plants the best. Now specifically, we ran a 2x3 factorial design, which means that we were looking at the differences between two independent variables. And those variables were lighting, so high light versus low light, and the other independent variable was substrate. So we were looking at the effects of three different types of substrates, specifically gravel substrate, montmorillonite clay, and the third substrate was Rockwool. Now I just finished harvesting all of these plants and I've ran all of the statistics, so we have the results for which method grew the plants the best. So we're going to dive deep into all of the results in this video. Now if you haven't seen part 1 of this experiment series, I recommend you go back and watch that video because it talks in a lot more detail about the experimental design and why we were running this experiment in the first place. Now in that video, you'll see that I did this experiment using two different types of plants. Obviously here we have the Anubias plants, but I also ran this experiment using cryptocorn plants, which as you can see behind me, the crypt plants are still in the setup, which means I haven't yet harvested and ran the stats on those plants. Now it took me a really long time to harvest these Anubias plants because we weren't just weighing the plants, we were also counting all of the leaves on each individual plant, as well as the number of shoots grown on each plant as well. And just the sheer amount of plants that we had in this experiment meant that it took a really long time to get through all of that, let alone preparing all of the statistics and the graphs. So for that reason, I'm leaving out the crypts from this video and we're just going to dive deep on the Anubias results, which, not to spoil anything, we do have some really incredible results that I'm excited to share with you all. In a follow-up video, I'm going to process the crypt plants and discuss the results, and we'll see if there were any differences between their growth compared to what we got for the Anubias plants. Really quickly, before we get into the results, I want to show an example of how I process these plants just so that all the terminology makes sense. So I've got an example Anubias plant here that I was just growing in a separate container outside of the experiment, but I'm going to go ahead and process this just like I did with all the other Anubias plants just to show exactly what happened. So as you can see here, we have our example Anubias plant that we are going to process, but we also have a few other tools on the table. So the first one, and probably the most important, is this pocket scale here. Now this scale measures to 2 decimal places, which is going to give us a really great precision for this experiment. And then I also have a small bowl that I'm just going to place on top of the pocket scale. The reason for this is because some of the plants that we're processing are quite large, and we want to make sure that all of the mass of the plant is being directed on the scale and not touching the table, which would throw off the measurement. Now we can easily zero that out, and then anything that we need to measure we can put into the bowl and get a precise measurement. Now on the right hand side here, we do have some sharp stainless steel scissors, and the reason for that is to help with cutting the roots of these plants. Now the reason why I'm cutting the roots of the plant is so that we can get a few different metrics that we can run stats on. Now obviously we can get the total biomass of the plant, which would include both the main plant portion as well as the weight of the roots, but by cutting the roots of the plants we can also just measure the individual plant biomass as well as the root biomass and see if there were any differences within these parts of the plants across our experiment. So this specific plant was growing in the montmorillonite clay substrate. So as you can see here, it comes out quite easily. Now the majority of the clay is still in the net pot, which is great because we need to separate it from the plant. But as you can see, the roots are quite extensive and there is quite a bit of clay here still attached to the roots. Now in order to get a fair measurement for the root biomass as well as the total biomass for the plant, we're going to need to remove as much substrate as possible. And for that, I found that agitating these plants in a bucket of water was a pretty effective method for removing the substrate from the roots. And just like that, with a little bit of effort, we can remove all of the clay substrate from the roots. And as you can see here, the roots are looking pretty healthy. They are nice and white, with the exception of the roots that were exposed to light, which developed a little bit of a green pigment to them. Now the first data point that we're going to collect is the total biomass, which is going to include both the roots as well as the main plant portion. So for that, we're just going to turn on our scale, very easily toss that in, making sure all the roots are in the bowl. It looks like this one is exactly 6 grams on the dot, so we would then record that in our Excel spreadsheet and then move on to another metric. So the next step in processing these plants is to actually remove all of the roots. So for this, I would use the scissors and cut the roots just at the base of the rhizome. Trying to get as many roots removed as possible. So as you can see here, we've got the clump of roots and the main plant portion. And so the easiest way to go about this next is to measure just the plant, and we're going to get the plant biomass. So as you can see here, the scale is reading 4.74 grams, and we'll then record that in our Excel spreadsheet. And now by taking the difference between the 6 grams and the 4.74 grams, we can now get the biomass of the roots, so we don't have to fill around with all of the individual roots and trying to fit them all in the bowl. So now that we've got all of the weight measurements completed, we can now set our scale off to the side, and we still have 2 more things that we have to measure. So the first one is number of leaves on the plants, which is pretty straightforward. However, as you can see, as the plant gets more and more mature, the leaves are quite densely grown into the plant. And so I find it does help a little bit to separate if there were any side shoots of the rhizome. We could separate that off and count them individually, just so that we're getting an accurate representation of how many leaves were on the plant. And lastly, the last thing that we were measuring is the number of shoots. So as we know, an Anubias plant grows through their rhizome, and over time, that rhizome can actually split and develop side shoots that grow into their own plants. And in fact, as a hobbyist, if you want to propagate these plants, it's a really effective method to cut off those side shoots. And you can actually plant those in another immersed setup or an aquarium, and it'll grow into its very own Anubias plant. So this is a very important metric, especially if you like propagating Anubias plants, because knowing if there's a method that grows a lot of side shoots is going to be directly proportional with the amount of Anubias plants that you can then go ahead and propagate. And as you can see, this plant actually does have a few side shoots that I can show you an example of. So obviously this end here is the main growing end, but we can see that just down here there was a side shoot that is starting to form, and I can go in with the scissors and actually remove that side shoot from the plant. So as you can see here, this little side shoot, it's not the most mature side shoot, but this would certainly grow in its own immersed setup and develop its own Anubias plant. And in fact, this plant actually does have a few other side shoots. So for example, there's an immature one just over here, and then on the underside of the plant, there's another one growing just right over there. Now both of these are pretty immature, so we'll leave these ones on the plant. Now after recording the number of leaves and the number of shoots, these are all of the data points that we collect from these plants, and everything else is processed in Excel and Python. Alright, so with the demo out of the way, now let's take a look at the results. So one key aspect of this experimental design is that we kept track of each of the plants that went into the tubs. And in fact, we even developed randomized planting charts so that the 3 different substrates were randomly distributed into each of the growing tubs. Now this allows us the opportunity to develop heat maps to see if there was any external environmental factors that cause the plants to grow more or grow less. So let's take a look at the heat maps for total biomass gained. So as you can see here in the highlight and the low light tubs, there are certain areas that appear to grow plants a little bit more than others. So for example, the outskirts of the tubs maybe didn't grow the plants as much as the inside of the tubs, and that may be due to light distribution. So because of the combination of lighting and the 3 different types of substrates, we actually have 6 different experimental treatments that we can compare to one another. So when we graph the average total biomass gained across all 6 treatments, we get this graph here. As you can see, there are some fluctuations in the amount of total biomass gained across all 6 different treatments. However, once we ran the ANOVA, you can see that none of these differences ended up being statistically significant, which is denoted by the letter above each bar. So when you're looking at these kind of charts, if the letters are the same, then that means those differences are not statistically different. If we were to have a different letter above one of these bars, it would mean that it is statistically different from any other bar that doesn't have that same letter. So there's one major fault with looking at the total biomass gained in this experiment, and that is because we can't control the starting weight for each plant that was put into this experiment. And what I mean by that is a plant that has a higher starting weight or is a bigger plant going into the experiment may have an advantage because it might have more leaves, or it might have more resources that could send out new leaves and propel the plant to grow faster than plants that are smaller in the experiment. So for example, if we were to start a new experiment and use this plant versus this plant clearly has an unfair advantage because it is simply a larger plant and it has more resources that might kickstart the growth and outcompete a plant such as this. So to account for this, we are able to actually use our data and calculate a new metric called the relative growth rate, which normalizes and takes into account the starting weight for each plant in this experiment. And that way we can actually compare the values between all of the plants in the experiment, and we don't have to worry about if one plant was larger at the start of the experiment compared to another. And the really interesting thing is when we convert the total biomass gained into the relative growth rate for total biomass, and we look at the heat map, we actually get a much more uniform growth across the plants. So as you can see, there are no more hotspots that appear to be growing plants faster than the others. There may still be an effect for plants that are closer to the edges of the tubs. They still may be getting a little bit less lighting, as you can see in this heat map, but overall things look a lot more evenly distributed, which to me tells me that maybe some of the hotspots that we saw in the total biomass heat map were actually due to those plants having a higher starting weight compared to some of the other plants. And next, when we compare the averages for the relative growth rate for total biomass, we can see things actually seem to be a little bit more even, and again, we don't have any statistical differences across any of our 6 treatments. Next up, we have root biomass gain. So as you can see with these heat maps, we get a very similar pattern to the first heat map that showed the total biomass gain. And that makes sense because typically if a plant is going to have a larger total biomass, then it probably also has proportionally a higher root biomass. And by looking at the averages for the root biomass, we actually do have some statistical differences in our results. When we compare the highlight Rockwool treatment to both the low light gravel and low light Rockwool treatments, those differences actually came out to be statistically significant. Now you can see this depicted in the bar graph with the letters above each bar. However, to help visually depict where these statistically significant differences lie, I've also prepared an ANOVA comparison matrix, which helps to visually depict where these statistical differences are. So as you can see here, with 6 different treatments, we actually have 15 different comparisons for each possible combination in this experiment. And so the ANOVA comparison matrix actually makes it quite easy to visually tell if any of those combinations ended up being statistically significant. So for example, if you follow the highlight Rockwool up to the comparison with low light gravel, you can see that that difference was statistically significant and ended up having a p-value of 0.0034. And as pretty standard with most scientific experiments, we're using a p-value of 0.05 to tell when something is statistically significant. And we just looked at the root biomass, so now let's move on and look at the plant biomass gained. A very similar trend can be seen in the heat maps that shows there are a few plants that had particularly good growth in this experiment. And when comparing the averages across all 6 treatments, again, we don't have any statistically significant differences. And similarly to the total biomass, we can actually calculate the relative growth rate for plant biomass. And we see that the heat maps are actually pretty evenly distributed, meaning that for the most part, the plants were growing pretty evenly no matter where they were located in the tubs. Again, when comparing the averages across the 6 treatments, in this metric, we didn't get any statistically significant differences. I was also interested to see if different substrates actually promoted more or less root growth in our Anubias plants. And so to evaluate that, I calculated the percentage of the plant weight that was made up of roots, and we can actually compare that across our 6 different treatments to see if there was any differences in our experiment. And so interestingly enough, when you look at the averages across the 6 different treatments, we do actually have a few statistically significant differences. So looking at the bar graph and the ANOVA comparison matrix, we can see that the Rockwool actually appears to grow a higher proportion of root mass in comparison to the other treatments, with actually the highlight Rockwool treatment having the highest proportion of root biomass. Now this difference would suggest that maybe Rockwool does promote a little bit more root biomass in each plant. However, I am personally a little bit skeptical with this, simply because this also comes down to how well I can actually pick out all of the Rockwool when I was processing these plants. And while I thought I was doing a really good job removing all of the Rockwool, there is a small chance that maybe I wasn't picking out as much Rockwool as I thought I was, and leaving a little bit extra on the roots actually swayed those treatments to artificially seem a little bit higher biomass compared to the other treatments. Nevertheless, I'm more interested in actually the plant growth as opposed to the root growth, and because we weren't seeing any statistically significant differences in terms of the plant growth, I don't really think that having more roots or less roots is really having an effect on the growth of these plants. Alright, so next up we're looking at the number of leaves that were gained on our plants, and this is where things actually get really interesting. So again, when looking at the heat maps, we can see that there's a very similar trend happening where there are a few individual plants that really excelled in this experiment. However, when looking at the averages across all 6 treatments, there are drastic differences that can't be ignored. So as you can see, regardless of the substrate that was used, the highlight treatment had much higher number of leaves compared to the low light treatment. And I actually mentioned in my last video that looking at the plants and how they were growing, the highlight bins seemed to have a lot more compact growth, and it seemed like they had more leaves on each plant, whereas the low light treatment actually visually looked like it had a little bit less leaves, but each of the leaves looked a little bit larger. And we can see here with the statistics, this is highly significant, and our ANOVA matrix shows that most of the highlight treatment options, again, regardless of the substrate, was statistically different from most of the low light treatments. The only exception there is the highlight gravel treatment was not statistically different from the low light clay treatment. Now again, we might have an issue here because this result doesn't take into account how many leaves each of the plants started out with. And as we know, a plant with more leaves starting out in this experiment theoretically should have an advantage because it has more leaves that could photosynthesize and produce more resources to grow the plant. So again, we can standardize this data and we can calculate a relative growth rate for the number of leaves gained and basically eliminate the starting number of leaves as a confounding variable when comparing across our treatments. Again, when we look at the heat map for the relative growth rate for number of leaves, we don't have as many hotspots and the growth across the board looks a little bit more uniform. And when comparing the averages, we still have quite a few statistically significant differences, although they aren't as many as before. So as you can see here, the highlight treatment options with gravel, clay, and rockwool were statistically different from the low light gravel and the low light rockwool groups. However, the low light clay groups didn't end up coming out being statistically different from any of the highlight treatments. And lastly, the final metric to look at is the number of shoots gained. So when looking at the heat map here, we can see that there are a few hotspots that had particularly high number of shoots that were grown on the plants. And more importantly, the low light treatment actually does have a few areas that really didn't perform well in developing new shoots, which is quite interesting to see. Now, when we look at the averages across all 6 treatments, there are major differences that we can see right off the bat. Very similarly to the number of leaves gained, we have a similar trend where the highlight options, regardless of the substrate, ended up having quite a few shoots that were grown on the plants, whereas the low light treatment didn't really grow as many shoots in comparison. And most of the comparisons between the highlight and the low light treatment options are statistically different. In fact, there's only one, which is the highlight gravel compared to the low light clay, that is just a marginal significance, which basically means that the p-value is close to 0.05 but didn't cross that threshold. And so for that reason, I'm calling that a marginally significant difference, whereas all of the rest are lower than 0.05, so we can conclude that that is statistically significant. So that was a ton of results to look at, but what does this all mean in terms of growing these Anubias plants, and which treatment was the best treatment for growing these plants? Well, for starters, when looking at the biomass that was gained in the plants, we didn't have a lot of statistically significant differences. Now, the reason why I think this was the case is because I ran this experiment for quite a long time, and typically when plants are growing in a confined environment, similar to our immersed setups, eventually they reach a point where they start choking each other out and they reach their maximum capacity. And at which point, when they reach their maximum capacity, they start competing with one another and the growth across the board slows down. However, the issue with running this experiment for as long as we did is that if one treatment ended up growing faster than the others and reached its maximum capacity, it's possible that the other treatment ended up taking a little bit longer time to grow, but ultimately ended up reaching the maximum capacity and resulted in no statistically significant differences across the board. Now, it may have been better if we wanted to map out the growth rate of the plants to have subsamples at periodic times between the start of the experiment and the end, and at that point we could see if there was any differences in the growth rate across the plants. So that may be an improvement and another experiment that we have to run in the future. However, there are still very meaningful results from this experiment, specifically when we look at the number of leaves and the number of shoots that were grown. it was very apparent that regardless of the substrate that we were using, the highlight option grew these plants much better in terms of the number of leaves and the number of shoots. As I mentioned earlier, if you have a way to grow the plant so that it produces a lot more shoots, you can actually cut those off and get more individual plants when propagating. So based on these results, I would conclude that the highlight treatment is by far much more productive than the low light treatment, and is personally probably the method that I'm going to use going forward with growing my plants. Now in terms of substrates, the results show that there may be a few minor differences across the 3 substrates that we were evaluating. However, I would say that there aren't any major changes in the way that the plants were grown that would lead me to believe that one substrate actually performed better than the other. In fact, knowing this, I would probably opt for a substrate that is either cheaper or easier to work with. So for example, the gravel is actually pretty clean when harvesting the plants in comparison to the clay, which produced quite a bit of a mess. And the Rockwool is a little bit more expensive. So I would say choosing whatever substrate is more convenient would be a more important choice, knowing that it's not going to have any major differences in the way that the plants grow. I hope you guys enjoyed this video and took something away from the results. If you like these kinds of videos, please subscribe to my channel. I've got a lot more of these experiments coming down the road, and I'm really excited to continue sharing them with you all. And don't forget, we also have the results coming from the cryptocorn plants that were grown in this same experimental design. So that video will be coming out shortly. In the meantime, please click through to this video right here if you want to see another experiment. And until next time, take care.